All right. Okay. So we'll start. Good morning, afternoon, evening. Hola, hello, bonjour, hola, and ni hao, konnichiwa, 안녕하세요, and hello all, and welcome and thank you and SynBYSS people for joining us the 49th seminar, and I call it on MIT Day. Today's seminar is very special to us. August 26th was our 1-year anniversary for this weekly seminar series, and it has been my great honor and privilege to see your excitement and support for over 1 year. I am scheduling for the 2023 season, and I hope to continue to chair this seminar in the following years. Additionally, of course, today is September 1st, sort of, the start of a new semester. And I would say, what else would be better than starting a new semester by listening to great talks? Okay, it is my tremendous honor to introduce, of course, very briefly, our pioneer speaker, Professor Greg Stephanopoulos. He does not need any introduction because we all know him. He is an MIT professor, a member of National Academy of Engineering, and most notably, the metabolic engineering father who has contributed to establishing the field for many decades. Personally, I had the honor to know him since 2005 when I studied my PhD program at MIT, and the most recently had dinner with him when I received the 2019 BMB Daniel Wang Award, and at the time he also received the Gadden Award for Biotechnology and Bioengineering. He's right now in Greece and beautiful island, I heard. And Professor Stephanopoulos, thank you so much for your time and support for young researcher and your contribution to metabolic engineering. And the virtual podium is all yours. And thank you again.
Thank you very much, Taesok, and also thank you for initiating this wonderful series of seminars that gives the opportunity to many people, mostly young people, to talk about their work and expose the great things they are doing to all continents around the world. I think it's a wonderful initiative, and it's been appreciated by many people, young and not so young. I have 5 minutes to say a few things about metabolic engineering, biofuels, and Ben. Here's the gist of it. Many of you, I'm sure, are familiar about great efforts that have been going on for the past 60 years to utilize renewable resources for the production of biofuels. The most notable such renewable resource is lignocellulosic material, the process by which people have attempted to utilize this abundant renewable resource is through hydrolysis to simple sugars and then using metabolic engineering to convert these sugars into a variety of fuel types and chemicals. This is great work. It should continue, no question about that, because it is aiming at the end of the day to utilize a great resource which is renewable, which is large, and which is going to play an important role in creating a circular economy and a future which is sustainable. However, I have to say that at the present time, sugars are expensive, and the efforts to produce sugars from lignocellulosic material have not led to a point at which we can see that process materializing in a reasonable period of time. Unfortunately, the last lignocellulosic biomass-based plant closed down a year and a half ago. This is a major blow to the efforts of this type. But again, this signals that we should not be expecting that resource to play a significant role in producing biofuels for many years to come. So if lignocellulosic material is out, if sugars are expensive, what is out there which can be used to supply, especially the liquid fuels that we need for many types of industries in the future? I'm referring here, electric cars are making advances and they are going to be using renewable electricity, but besides electric cars, we need liquid fuels to fuel the aviation industry, the trucking industry, and the shipping industry. So we are going to need liquid fuels for many years to come. Where are these liquid fuels going to come from? Well, another source for this can be gases. It can be gases of the type of carbon dioxide reduced or activated by a reducing agent such as carbon monoxide or hydrogen. The product of this can be formic acid, it can be acetic acid, or other types of chemicals. And then one can apply metabolic engineering to convert these chemicals again into a type of biofuel or into a type of chemical. There are many interesting issues associated with the utilization of gases, many of them of biochemical engineering nature. We need to worry, for example, about the transport rates of these gases from the gas phase to the liquid phase. We need to be concerned about utilizing electrons of different types in fixing CO2 and making products out of that CO2 fixation. And the combination of biochemical engineering and metabolic engineering, I think, is at the root of utilizing and eventually commercializing this technology. The background of the individual who is going to do that in the most effective manner is definitely of the type of a chemical engineer with a strong foundation in genetics and understanding of all types of biochemical engineering, but at the same time understanding how molecular biology can impact this field by generating all the different types of products and molecules that we need in today's economy. And there is no better person to do this kind of work than the speaker of today's seminar, Ben Woolston. Ben has an outstanding background in chemical engineering, biochemical engineering. He has demonstrated his ability to engineer microbes and create incredibly efficient cellular factories for making different types of products. He is now pursuing a number of directions in the production of fuels and chemicals. I know some of them. I'm going to say nothing about any of them because some of that is privileged information. So Ben will decide how much of that he wants to share with our audience in his talk today. It is really a great event that he is the first speaker of the series for this new academic year. And I'm really thrilled that I have the opportunity to introduce him to the audience of this series and look forward to his comments and his presentation.
Wonderful, thank you. Absolutely, I agree with you. So, I mean, gas will be our future source for making chemical and liquid fuels. And I believe, yes, Ben is the right person to do that. So, I mean, I thrilled as well because with your vision and then Ben's implementation in the field, I believe we could solve the problem, including climate crisis and sustainable, production of many chemicals. So thank you so much, Greg. I mean, this is absolutely brilliant and amazing talk. Thank you. Okay,
My
so
pleasure.
yeah, thank you. Thank you so much. Okay, so now the main speaker of today with a longer introduction. Dr. Ben Woolston joined the Northeastern University Chemical Engineering Department as an assistant professor in January 2020, probably the most challenging year because of COVID, as you know. He obtained his bachelor degree in chemical engineering with honors at the Pennsylvania State University, where his undergrad research won the AICH National Student Paper Competition. So we already see, and he's the potentially rising star. As an NSF graduate research fellow, another very competitive, the fellowship. He received his PhD in chemical engineering in 2017 from MIT under the guidance of Professor Stephanopoulos, today's speaker. During his PhD training, he focused on the development of genetic tools to enable metabolic engineering in anaerobic CO2 fixing estrogen and the establishment of methanol utilization pathway in the model organism E. coli. His postdoctoral work was conducted in the lab of Professor Emily Barkscott at Harvard University, where he studied microbial metabolic pathways and enzymes that contribute to the stability of health-associated lactobacilli in the human vaginal microbiota. At Northeastern, his research program combines approach from his previous research training in metabolic engineering, synthetic biology, biochemistry, and microbiology to engineer microbes for biofuel, biochemical production from single carbon feedstocks, and as diagnostic and therapeutics in the human gut microbiota. He is the winner of the 2020 J. Bailey Young Investigator Award and also the 2021 BNB Daniel Wang Award, to name a couple. He's truly a rising star with huge potential. And, I feel like, he's already a tenured professor when I prepare this, introduction. So Ben, thank you so much for your time today, and please take it away. And then I was going to say, if you are the tenure letter writer, I believe you should write a fantastic letter for him in the future. Thank you.
Oh, thank you very much, Taesuk, for that very kind introduction. With your words and Greg's words certainly put a lot of pressure on this presentation. So hopefully I can deliver. So just as we get started, can I just check that everybody can see the slides okay? You're seeing the right version. Fantastic. All right. Well, again, thank you for the invitation to give this talk. Very much looking forward to sharing some of the initial work that's been coming out of my lab. As Taesuk mentioned, I started in January 2020, which was certainly an interesting time to launch an independent lab. But I think we've made some good progress in that time, and I'm happy to share that here today. So the talk is biofuels and bioproducts from methanol via the Acetogen eubacterium limosum. So we're going to be talking about C1 metabolism and what we can do with some of those gaseous molecules that Greg already introduced to us. So before I get started into the technical details of the talk, I just wanted to give a little bit of background about my lab overall. So as Greg and Taesuk mentioned, what we're really trying to do is use chemical and perhaps even biochemical engineering principles to understand and manipulate microbial metabolism for human benefit. So all the projects in my lab are focused on learning something about bacterial metabolism and applying that, either making new pathways or making sensors, these types of things, to solve what we think are kind of some of the biggest challenges facing the world right now. And so we've divided that largely into the production of renewable biochemicals and efforts in the human gut microbiota. So I'm only going to focus on the first of those here today, but I'm always happy to talk more about some of the work we're doing in our gut after this session if people are interested. So when we think about renewable biochemical production, really, I think actually Greg did a fantastic introduction to this topic, which is we are going to need liquid fuels for transportation and shipping in the long-term future. And also we're still going to need carbon-based materials, right? Bioplastics and these types of things for a long time to come, right? And what we as metabolic and biochemical engineers are trying to do is to produce these from renewable resources, right? Using all the benefits of biology, ambient temperature and pressure, and all of these things that we as a field have talked about for a long time, right? But the challenge with this really is that if you look at most of the processes out there today, most of the commercial processes, still we are using food crops as the kind of ultimate source of the sugars that are feeding to microbes, right? So if we think about bioethanol in the US, that's primarily derived from corn. In Brazil, that's from sugarcane. And I would argue that if we want to be truly sustainable and truly renewable, we need to move away from food-based crops as our main source of carbon for fermentation. Before I get started, I just want to do a quick shout out. We are actually actively hiring postdocs into this area. So if you are interested by any of the things you hear about today, please reach out to me and I'd be happy to discuss. So if we don't want to use food crops, what do we want to use instead? Greg has already introduced lignocellulosic biomass. This is an abundant, widely available resource. But as mentioned, converting that to accessible sugars is still technologically difficult and expensive, right? And there's a lot of, I think this field really knows the challenges associated with that. There has been, over the last 20 years or so, interest in photosynthesis. So using algae or cyanobacteria, this would potentially have the benefit of not needing any kind of arable land. But at the same time, when you look at the efficiencies of photosynthesis, right, which are anywhere between the 0.1% to maybe 1% at highest, compared to, for example, photovoltaics, which are now reading up to 20%, this becomes not a particularly efficient process and therefore very difficult to scale up to the levels that we'd actually need in order to produce the molecules we want at scale. So if these are not the options, what are? Well, what we are very excited about is the idea of using single carbon substrates, right? So over the last 10 years or so, there's been a huge amount of interest and a huge amount of investment from the DOE and elsewhere, looking at molecules ranging from CO2, right? So the ultimate kind of waste gas that we would like to mitigate, to things like carbon monoxide, formic acid, methanol, and methane, right? So many of these are readily available. For example, there is a lot of methane being flared from abandoned wells in the US. Carbon monoxide and formic acid are starting to be produced using electric catalysis directly from CO2. So this is really exciting now, using renewable electricity to produce C1 intermediate molecules. And also methanol, which can be produced for a number of ways that I'll talk about in a little bit. So we think there are real promises of using these compounds. They don't compete at all with the food supply. In many cases, there's an established infrastructure that we can use to get these around where we need them. And almost all of these can be produced renewably, right? And some directly from CO2 and some from other waste resources. So I think the ability to use waste to valorize materials which right now are just being burned or not dealt with particularly effectively as a way of producing the fuels and chemicals that we need will really lead us to that circular bioeconomy. So this all sounds great, but what are the challenges? Well, really it comes down to the fact that for the microbes that can consume these substrates, this is generally not things like E. coli, not things like Saccharomyces cerevisiae. These tend to be less understood organisms. In some cases, we still don't actually know all the details of the metabolic pathways, right? And I would argue if you want to engineer a microbe, you really should understand its central metabolism before you try to do that. And then even if we do fully understand the metabolism, a lot of these microbes don't have great genetic tools. We can't do all the fancy, amazing things that we can do in E. coli and other microbes. Right? And so this is really in this area the challenge that my lab is working on, right? Can we develop this fundamental knowledge? Can we advance these genetic tools to the point that we would be able to actually do this with these types of microbes? So today's talk, obviously, I'm not going to talk about all these different single carbon substrates. Greg mentioned carbon monoxide coming from CO2. We're going to do another talk on that later, perhaps, but today I want to focus on methanol, which is a substrate I'm particularly excited about. Let me tell you why. So methanol is particularly attractive feedstock. If you look at this map here, this is just showing the various plants that exist throughout the world that are commercially producing methanol. Much of this is currently fossil produced, right? But some is actually what we call biomethanol, right? So this is now produced from renewable resources. So from a biochemical standpoint, when we compare glucose to methanol, for example, methanol has about 50% higher energy density, right? So the amount of energy per carbon input is much higher with methanol, and yet the price is about the same. So if we want to make fuel, which is always going to be a lean industry, thinking about making the best use of that carbon, having something that starts at a high energy density is a good thing to have. We're also seeing an expanding capacity of renewable methanol with predictions of 50 million metric tons per year by 2050. Let me give you a couple of examples of plants that are already online that are kind of exciting. So one is Carbon and Recycling International. Let me just turn my laser pointer on. So this is actually based in Iceland. They use geothermal energy, which they have in abundance there, to produce electricity, which then uses hydrogen to fix CO2 directly into methanol. Right? So we now have renewable methanol coming essentially from abundant free energy. Right? And then closer to home, at least if you live in the US here, Enerchem, which is based up in Alberta, Canada, they're using municipal solid waste. They're gasifying that to syngas and then reducing that to methanol. Right? So these are two actively being developed plants for producing renewable methanol. And the costs, they're still slightly higher than for regular methanol, but they're coming down. Right? And so we see this really nice potential for methanol to be a renewable resource at reasonable cost in the near future. Compared to some of these other C1 compounds, Greg mentioned the challenge of mass transfer, right? So arguably the field of biochemical engineering got really big because of engineers figuring out how to deliver oxygen to reactors, right? For growing cells to make penicillin and these kind of things. Well, it turns out some of these gases, hydrogen, carbon monoxide that people want to use, are even less soluble in liquid than oxygen, right? So this becomes a major challenge to actually get the gas to the microbes where they need to metabolize it. Methanol, though, is a liquid, right? So we can just mix it directly into the fermentation broth, and we don't have those same challenges of mass transfer. Okay, so with that background as why we focus on methanol, this brings up what I like to call the metabolic engineer's dilemma, right? So I mentioned that there are not that many microbes out there that we work with on a regular basis that use methanol, right? And those that do exist, we often don't have the best genetic tools. So you as a metabolic engineer looking at this problem, have a couple of options. One is to say, okay, maybe I can take a native methylotroph, so methylotrophs are what we call organisms that grow on methanol, and maybe I can develop those genetic tools, right? And then once I have those genetic tools, I can figure out metabolism and start engineering them to produce value-added products. Right? That's one pathway. The other pathway is we can go into what's called synthetic methylotrophy. And this is to say, well, maybe we understand well enough the pathways that are involved that we could take them from a native methylotroph. We could figure out all the enzymes necessary in the pathway, take those genes, put them in something like E. coli, and now we engineer E. coli where we've got a wealth of knowledge from the last 60 years working in it, and basically drop in a methanol pathway to allow E. coli to produce products from methanol. Right? Now, both of these approaches have their merits. We actually wrote up a little discussion of this that was published recently. I encourage you to take a look at it. To me, the takeaways come down to this, what I'll say on the next slide, which is, just presenting a simple example here. Fairly recently, we had a report of finally, after about 7 to 8 years trying, a group managed to engineer E. coli to grow on methanol as a sole carbon source. Right? And the stars around here just show all the different mutations that had to be made, all the evolution that was done in the strain. And after all that, was amazing effort from Jim Liao's group, terrific paper, really good results. But still, the maximum OD these cells get to about 1.9 with a really slow growth rate and not a great yield. Right? So this is after a huge amount of investment, a lot of work. Let's compare that now to Eubacterium limosum, which is the organism I'm going to talk about today. Without any engineering, right? So just natively what it does, we see in our lab a maximum OD of close to 3, a similar growth rate, right? And a much better yield, right? So I would argue that I think it's really interesting to look at synthetic methylotrophy. We learn a lot about metabolism when you try to build in another microbe. But I think there's this huge untapped potential to go after microbes that natively have the ability to want, and then figure out how to engineer them to make the products and fields of interest. Right? I think if you'd asked me this 10 years ago, I wouldn't have said the same thing. I think what has changed, though, is our ability to rapidly develop genetic techniques for new microbes has and continues to get faster, right? So with sequencing costs going down, synthesis costs going down, companies like Ginkgo, places like the Agile Biofoundry, JGI, is making it much easier to go out and develop these genetic tools so that you can start with something that does more or less what you want and then figure out how to engineer it later, right? And so because of that, we're really excited about using native microbes to look at their methanol metabolism. Okay, so which native methylotrope to look at though, right? So it turns out there are many different pathways in nature for using methanol. And I really like this way of looking at it, which comes from Aaron Bart-Evans' group a few years ago. As engineers, metabolic engineers, there are a few metrics we really care about, right? Titer, how much you produce, rate, how quickly you produce it, and yield, right? How efficient were you at using the substrate? So what this graph here is doing is showing on the x-axis the energetic efficiency. So how much of the energy of the substrate that came in was actually captured in the product at the end of the day, right? So obviously this can't go any higher than 100%. So that's essentially our yield metric, right? And then electron consumption rate, think of this as productivity, right? So this table is actually designed to allow you to look at multiple different substrates. But essentially, this is the rate at which you're consuming the C1 feedstock, right? So what you'd like to have is something that's very energetically efficient, so over here on the right-hand side, and also fairly productive, right? It can take up the substrate quite quickly. And so when you then put all the different native methanol utilization pathways and other CO2 pathways, etc., on this plot, what you consistently see is that these microbes called acetogens, which I'll go into in just a minute, consistently fall at the top range here. Very high energy efficiency. These are microbes that have evolved to live kind of a thermodynamic limit of life and kind of scrape out all the energy they possibly can from the substrate and be very competitive. But they still have fairly good specific uptake rates of the substrate as well. And so because of this, we're really excited about acetogens and using them for methanol metabolism. So what does methanol metabolism look like in an acetogen? Well, the central aspect of this is what's called the Wood-Ljungdahl pathway. You can think of this essentially as a linear pathway that takes two molecules of CO2 and converts them into one molecule of acetate. Right? So I won't go into all the gory details here. This is on YouTube, so you can look at it later. And basically, what happens is methanol feeds into one side of this pathway. Some is oxidized to provide, oxidized this way to provide reducing equivalents for growth, to provide some ATP. And then we have CO2 being fixed on the other side to then produce acetate. So the main takeaway from this, don't worry about the details of the pathway. The main thing is to look at the overall stoichiometry, right? So we're converting methanol to acetate. But what's really exciting to me about this is not only are we converting methanol, we're also fixing CO2 in the process, right? So this is now a potential benefit of acetogenic methanol metabolism, where as well as the benefit we're getting from our renewable carbon source substrate, potentially we can also fix some additional CO2 as we go, and that makes our process even better from a climate mitigation standpoint. So this is kind of the central pathway that underlines acetogen metabolism. One of the challenges working with acetogens is these are obligately anaerobic microbes. A lot of these enzymes are very oxygen sensitive. And if we look within the acetogens, basically the reason we chose the limosum, it grows on all these different C1 substrates very well. And when we started, it did have at least some genetic tools, some plasmids out there. Some people had managed to engineer some pathways in. So we thought this was a really good starting point for the work we wanted to do. All right, so with that kind of as a background, what I'm going to spend the rest of the talk focusing on is what we have actually now done or are continuing to do with this microbe. So the first I'll go through is work by one of my students, basically expanding this genetic engineering toolbox, taking these fairly basic tools and now making it much more robust, allowing much better types of metabolic engineering. And a really interesting kind of more fundamental look we're taking into methanol assimilation and the pathways involved, to see if we can actually boost the growth rate even more and make these processes more effective. And so we'll spend a little bit of time on both of those. Genetic tool development, this has all been, I have to thank the student that actually did all the work. This is Patrick Stanford, just about to start his 3rd year in my lab. This work recently came out in JIMD, so I encourage you, if you are interested in details, then take a look there. And basically, this figure just sort of summarizes all the work he did in a fairly short time, right? So new antibiotic resistance markers, new promoter libraries, some interesting work looking at the oxygen tolerance to try to make our transformation process faster, and some other things which I'll go through. But the kind of the point of showing this is, I think this work really nicely highlights how quickly we can now start to build out better genetic tools for native sort of non-model microbes now. So the first thing he looked at was optimizing transformation conditions. So basically looking at different voltages that we could use, different types of converts, how you prepared the different cells, how much DNA you added. And now we have transformation efficiencies. We measure these in number of colonies per amount of DNA that rival the best cross-trivial transformation protocols that are out there. One of the really interesting things we found working with limosu is, although it's a strict anaerobe, it can actually tolerate oxygen for a surprising amount of time. So this was a very simple experiment where we took the cells out of the anaerobic chamber, put them in just a buffer, PBS, and then took them back into the chamber over at different times and plated them out to see how many had lost viability. And what we found is we can actually take Eubacterium limosu out of the chamber for about 4 hours with no loss in viability. Right? So this is great when we think about some of the workflows we'd like to do. If you think about fax sorting, right? If you think about just being able to work on the bench a little bit with these microbes, there really are challenges working in the anaerobic chamber all the time. And so the fact that this is a little bit oxygen tolerant, it really helps us with this. We were able to take advantage of this and actually show that we can actually do our electroporation on the bench of these cells, right? So when we compare to our standard protocol where we prepare the competent cells in the chamber and then shock them in the chamber, this is sort of always our best result. But we can actually take them out of the chamber and shock them on the bench before putting them back in for the rest of the transformation. So again, this just starts to speed up the rate at which we can do some of this work because we're not quite as tight in the anaerobic chamber. I think there's also some really interesting basic questions about how these microbes tolerate oxygen, why it's different. Some citizens I've worked with are much less tolerant. Right? So I think some interesting questions there. We also wanted to do kind of colorimetric plating. So many of you here are probably familiar with blue and white screening. Well, that doesn't work under anaerobic conditions, but we have what we call black and white screening. So this compound S-gal is a little bit like X-gal, but basically once it's been cleaved by the enzyme of interest, if you incubate this with iron-3, you kind of get this black precipitate showing up. And so when we transform cells with a plasmid expressing the BGA-B gene, we can actually see this, right? So now we can also do colorimetric screens for things like promoter strength and that kind of thing, all in the anaerobic chamber. One major limitation of working with elim when we started was there are only 2 different antibiotic resistances. And so when you start to want to do more advanced things in the genome, kind of swap genes in and out and those kind of things, it's really nice to have a couple more markers to work with. And so we did some screening and we were able to find tetracycline as another very good antibiotic, identified resistance markers that we could use, as well as ampicillin. So we've actually doubled now the number of antibiotic markers we have for this strain. Finally, we started looking at promoter libraries, right? We'd like to ultimately, with engineering pathways, be able to control genes at their expression level. And so we have a panel of promoters that we're looking at here, ranging from strong constitutive promoters that work in other Clostridia to looking for really stringently controlled inducible promoters. And so this is basically work we did, sort of showing now that we have quite a nice dynamic range of expression level that we can play with. One of the really interesting things that came out of this, look at the left-hand side of this, is we found that if we added, as we were doing these tests of different antibiotic concentrations with the cells, as we added more antibiotic, we actually saw the copy number of the plasmid going up, right? So in that, we had this interesting idea of, well, could we use this to give us further gradation in our gene expression, right? Could we essentially bolster expression from a strong promoter by making sure there was more of the plasmid around just by dosing in more antibiotic? And this actually worked really well, right? If you look at this NIF-JP promoter here, for example, right? When we have a higher concentration of the antibiotic, right, we get almost double the expression level. And so this gives us another way of kind of tuning expression without having to go in and make more genetic constructs. Right? This was kind of interesting and a neat thing to have in our back pocket. So, that genetic tool development work continues. We're now looking at more advanced techniques, things like CRISPR, and other kind of genome rearrangement type of things. But we have a good start and can now really kind of launch into some of the metabolic engineering work that we'd like to do. So I'm going to switch gears here and tell you about another project going on in the lab. This is being led by Katie Hoyt, who's just starting her 4th year in the lab. And this comes from a really interesting observation from a group in France in the early 90s, right? So I mentioned that the specific uptake rates of methanol in acetogens were just about as high as they are in aerobic methylotroph, so competing microbes that one might think about for developing these processes. And that's true. But one of the downsides of these acetogens is their ODs don't really get that high on methanol, right? So we see 2 to 3 highest, and the growth rates are quite slow. Right? So if you look, the doubling time on methanol is about 8 hours. The growth rate on glucose is much faster, right? Now, this group a few years ago did this really interesting experiment growing on a combination of methanol and glucose. Right? So this was done in a chemostat, so you could keep the glucose concentration low enough so you didn't have dioxy shooting in, and you actually had the strain growing on both substrates simultaneously. And what's really interesting about this is it actually kicked up the growth rate synergistically, so higher than was achieved on either substrate by itself. And this is really interesting because we tend to think of glucose as a preferred carbon source, but somehow by having 2 carbon sources, we can actually get better growth than on either one individually, which implies some kind of metabolic rearrangement, something in central metabolism has been completely changed to allow this to happen. Right? And so you could think about this from a practical standpoint of maybe this would allow you to get kind of higher cell titers, right, in your reactor, so you could then produce faster from methanol, right? Remember that ultimately what we care about is the absolute rate of methanol uptake. So that's going to be a combination of the specific uptake rate, so how fast an individual cell is doing it, and how many cells there are in the reactor. So we can boost the biomass in the reactor, we can boost the overall methanol productivity. Right? So that was interesting to us by itself. Even more interesting, though, was to look at the measured methanol uptake rates, right? So normally methanol is taken up at about 9 millimole per gram dry cell weight per hour. But when you add the glucose, the methanol uptake rate increased. So this kind of surprised us. I'm not aware of many examples of this, right? Where somehow adding a little bit of glucose to the culture is not only just sort of improving biomass because glucose is a better substrate, but it's doing something that actually improves the cell's ability to metabolize methanol. So we looked at this and we were really interested in this, essentially because the idea came with, well, perhaps if we understood how this works, if we understood why glucose is giving this benefit to methanol uptake, if we could figure that out, perhaps we can leverage that through some additional metabolic engineering, genetic engineering, to get strains that grow better on methanol, right? So we could essentially use this to make our ultimate bioprocesses better. So we wanted to do a lot of experimenting with this, but one of the challenges working with anaerobes, especially if you want to do continuous culture or fed batch to make sure you're balancing the concentrations of different nutrients, is doing all this in batch culture is very slow. You have serum bottles. It's hard to get really high resolution data. And you could put it in a bioreactor, but as an academic lab, it's hard to have enough bioreactors to kind of do the kind of screening we wanted to do. So what we wanted before we even started this work was essentially a high throughput mini bioreactor platform so that we could test all these different conditions, different methanol feed rates, glucose feed rates in parallel, right? While getting kind of constant OD monitoring and those kind of things. So we were inspired by work that came out of Mo Khalil's group back in 2018. And many of you may have seen this in this community, the Evolver platform that they put together, which is a really nice, low-cost, essentially DIY system that allows you to grow 16 different cultures in parallel, all with individual optical density measurements, temperature control, stir rates, in either batch, fed batch, or continuous culture. Right? So this is a really nice platform. We thought this would be great for the kind of screening work that we wanted to do. But the challenge is this was all developed for aerobic microbes. So the first thing we did was basically adapt this to work for our anaerobes, which are oxygen sensitive. So we did a lot of investigation of finding different ways to kind of close the caps. to make sure they were really anaerobic, that no oxygen was coming in. We focused on using tubing that had very low oxygen permeability, so we could do all of this out on the benchtop. And what ultimately arose from this, and this should be coming out fairly soon, hopefully, is now we have this really nice setup where we can do, so here are evolver vials, here are the actual cultures themselves. We're actually doing anaerobic fermentation on the benchtop, right? With feeds coming in of the different substrates, right? So this is a really nice platform for kind of the screening that we want to do to figure out this substrate co-feeding effect. So just some starting data I'll show you from this. So we were able to recapitulate the result from the literature and show that, so here we have, in blue is just cells growing on methanol, green is cells growing with methanol and that glucose feed. We set this up with a limited feed, so the cells were always glucose limited and had abundant methanol, right? And we saw exactly what we expected to see, right? So we see a significant increase in the growth rate, right, especially early in fermentation. And we also see an increase in methanol consumption compared to the control case. When you do the math out, that actually turned out, at least early in the culture, to be about an 80% increase in the specific methanol uptake rate. So we were able to recapitulate what had been seen. What was really interesting when we started looking at the products that Limosu usually makes, this is acetate and butyrate, there actually wasn't much difference. Right? So what this implied to us is that even though all this additional carbon was coming in, they were channeling most of that to biomass and not to the products. So this, I think, you'll see this a little bit later when we look at the labeling results too. All right, so we've been able to recapitulate what we've been found before. This is always a good first step, especially for a first PhD student to do. Now that we had this, we wanted to try to figure out, well, how does this work? From a fundamental metabolism point, what is changing that's allowing this increase in the specific methanol uptake and the specific biomass production rate? So one of the tools we have that we really like for doing this thing, especially as we're looking at multiple substrates coming in, is to think about using isotopically labeled substrates. So then we can basically feed a labeled isotope, and then later we can bust open the cells and measure different metabolites by mass spec to see basically where did different substrates go within central metabolism, and then from that kind of rebuild the picture of the different fluxes internally. But to do 13C label experiments, you really need to have a defined media, and we've been working so far on a medium that had yeast extract. So we did some experiments to look at what could we leave out of the media, and the kind of the summary of all of this is we were actually able to leave out the yeast extract and still see the benefit from co-feeding. Right? So this was really nice. It meant that we could start thinking about 13C isotopic enrichment experiments. So that's what we did, and don't pay too much attention to this slide because obviously this is a huge amount of data, and we actually only fairly recently got this data. So all unpublished for now, but we're in the process of writing up. Basically what we did is we looked at under either methanol growth or co-feeding growth, where we have the glucose, what happens to different isotopic traces that we add to the medium. So methanol obviously is one that we can label as a primary carbon source. Acetate we also label. We have some acetate in the medium that seems to help with the growth of Limosu. And then we can also label the glucose that's in the feed that's coming in through the pumps, right? And essentially by looking at all these data and then what we look at is we look at the amino acids in the biomass that are produced by the cells. We hydrolyze them down and we can look at them individually on a mass spec to see the enrichment of specific amino acids over time. And since we know in most cases we have a good genome model of this microbe, from which metabolites those amino acids come from, we can use that and go backwards and kind of infer the actual fluxes within the cell, right? So again, a ton of data here. We're still kind of working through it, but I'll just point to one particularly sort of interesting finding that we have so far from this data here. So one of the things we were sort of curious about was, okay, ultimately what we really want is for our primary substrate to be methanol, right? We're trying to convert methanol into products, not glucose and methanol into products. So we were really interested in, well, where is the product actually coming from, right? Is it coming from methanol or is it coming from glucose? And so here we, as well as looking at the amino acids, we also looked at the products that are being made. So you can derivatize acetate and butyrate and then run them on the mass spec and do the same kind of analysis. And so here we're looking, remember green is labeling data from glucose, right? It was interesting that early in co-feeding, right? So right when we start adding the glucose, still most of the product is unlabeled, right? So still most of this is actually coming from methanol, right? And then if you do the inverse of that experiment and you label the methanol, and you look at the results, obviously during methanol, a lot of this is labeled. Most of the unlabeled product is coming from the CO2 that's also, remember, required for this process. And actually, when you start the co-feeding of glucose, you actually see increase in the labeling of acetate. So they're actually increasing, incorporating more of the acetate is actually coming from methanol in this case, right? So kind of the main takeaway here at this point is that, you know, even though we're feeding glucose, we think most of the glucose flux is actually going towards amino acids that derive from glycolysis and from basically above the conversion from pyruvate to acetyl-CoA. So most of our product is still coming from methanol, right? So what this suggests to us is that, okay, we can probably get by with a very low glucose feed and still have most of our product coming from methanol, which is ultimately what we want to do. So obviously there's still a lot more we can learn from that labeling data. We're moving toward doing this in continuous culture and doing a full rigorous metabolic flux analysis. And so stay tuned for that. And again, ultimately the goal here is kind of twofold. One is to figure out how this mechanism works. I think it's really interesting that one substrate can improve the uptake of another substrate. And then eventually leverage this for producing chemicals directly from methanol. And so we've got some work we're actually putting in pathways for different things we're interested in here. All right, so I'll stop there and just kind of summarize, because I can see we're getting to the end of the time. Hopefully I've convinced you that methanol is an attractive feedstock for biofuel and biochemical production. And especially I hope I've convinced you that the acetogenic microbes are really interesting. You know, even though they are, you know, kind of non-model organs, we don't have as many genetic techniques. It's getting much easier now to build out those systems. So I think I really encourage, especially young people here looking for what to work on, you know, don't feel limited to just kind of the classic platforms that we think about, right? Go out and find interesting microbes that do interesting things. It's not as difficult now as it used to be to you know, bring them into the lab, domesticate them, and develop these tools. Everything you've seen in the last two slides was really done over the last 2 years by 2 PhD students in my lab, right? So we really can do a lot in this space now. So specifically, we've expanded these genetic tools. We've got this really nice low-cost, high-throughput platform for parallel anaerobic fermentation. This is great for kind of continuous reactors and all this kind of stuff. This really simplifies working with anaerobes. All the modifications that we made here, we're going to publish this. It'll be completely fully released, and everybody will be able to use this. So if you're interested in setting up something like this in your lab, if this would be helpful, feel free to reach out, and those details will be available. And I think the way we're kind of going with this is using these isotopic traces to figure out exactly what's going on with these rearrangements that underlie this really interesting co-feeding phenomenon. So with that, I'll acknowledge the students that actually did the work. Most of what you saw here was from Katie and Patrick. I'm hoping we get to the point soon where we can take lab pictures where everybody's not standing 6 feet from each other. Also have to acknowledge my collaborators, Mo Khalil, who has continued to help us as we develop this Evolver platform in our work, my active collaborators who are helping with some of the genetic tool development and some of the other work going on in the lab. Primarily ARPA-E for funding, as well as Northeastern startup funds. Thank you very much for your attention. Tayso, thank you so much for the invitation to be here today. It's really exciting to share our work. And I'll leave you with this final thing. I sort of highlighted this already. We are recruiting, so we're actively looking for a postdoc candidate to start as soon as possible, basically. So if any of the acetogenic work, the anaerobic gas fixation sounded interesting to you, that's actually a joint project between my lab and Greg's lab at MIT. So you could even get to work in both labs. They're actually very close. So here we are down at Northeastern, and MIT is just over here across the bridge. We've got some fancy kind of stuff we've set up for gas mixing anaerobic fermenters. Do a lot of work in anaerobic chambers, including anaerobic biochemistry, which I didn't talk about today. So yeah, we're always looking for new people to join the lab. Check us out on the web, or feel free to get in contact with me directly. All right, thank you very much. I'll stop there, and I'm happy to take any questions.
Oh my God, amazing talk. And I echo you one more time. Please apply for this position. Why not? This is the joint position between Greg Stephanopoulos and Ben Woolston. I would say, metabolic engineering father and, the rising star, I mean, already risen, I believe. So am I eligible to apply for that position? Okay, so fantastic. I wanted to check. Okay, I want to ask the question. So this is a fantastic. I'm very excited. Even though I'm not doing any gas related work or C1 thing. One thing I wanted to ask is, what would be your kind of perspective or vision in terms of commercialization of entire process? Is it 5 years from now, or 10, or and then can you give what is the hurdle, what is the challenges and opportunity out there?
Yeah, great questions, and we think about this a lot in my group. So there is some stuff that's really starting to happen now, and I would point to LanzaTech as kind of the prime example of this, right? So they've commercialized the process where they take a steel mill off gas. This is primarily carbon monoxide, carbon dioxide, and then some hydrogen. And they feed this to their microbe, which makes ethanol, right? And then that ethanol can either be fueled directly, or it can be upgraded to kind of jet fuel and these kind of things. And so that, there are plants all over the world doing this now, which is really exciting. Yeah. The work with things like methanol, formic acid, that's a little bit further behind. Part of the reason for that is, as I mentioned, there's different ways of getting to those. I think one of the most exciting ways is electric catalysis, right? So you could take a CO2 feedstock from something like a power plant or some other kind of concentrated point source, and then catalytically convert that into one of those reduced C1 intermediates, right? That technology is still being developed. Formic acid and CO are reasonable at the moment. The Faraday efficiencies are okay. And there are a couple of companies doing this now, actually. So a company whose name I'm blanking on right now, who's developing the CO technology. And I think one thing to highlight, so the DOE maybe a year and a half or so ago, through BETO, put $50 million into research to actually show that process at scale on the back end of a yeast bioethanol plant. Right? So the idea was that yeast bioethanol produces a ton of CO2. Could you then convert that into a C1 feedstock? So in the next couple of years, there are going to be demonstration facilities in the US, converting to that C1 feedstock there. I think that's still a long way from commercialization, but I think it's a great first step. If I think about some of the things I hear when I talk to potential investors, right? One of the ones they say is, well, can we scale your strain? We've never worked with limosu before. How difficult is that going to be to do? And it's a great question, right? Scale up is not easy, but I think it's also the kind of thing where until you do it, you won't know, right? So it's sort of a bit of a chicken and egg type problem there. I still think we've got a lot of work to do to really get to the type of rates and yields that we need with these microbes. But I think those first steps are there. And I think, if we can bring in kind of the big guns, right? So your Ginkgos, your ABFs, and companies that have got really good at kind of doing this large throughput strain building, I think that will help get us there. So yeah, some of the gas fermentation happening right now, methanol, formic acid, 10, 15 years, maybe even less. I think, it's becoming, what's really exciting to me is seeing actually like first year undergraduate students coming to my lab who are really interested in actually solving the climate challenge, right? There's a real impetus that people are really motivated right now to do this. And I think this is one of the central technologies that will help with that. So I see, I see a lot of buy-in, which is really great.
Yeah, absolutely. Yeah, amazing. Yeah, so I'm so excited, and then you mentioned multiple important point, during this answer. I mean, LanzaTech doing fantastic. Their technologies, amazing. The Nature Biotech paper recently published, that's the beautiful. And also you mentioned at the end of your answer, you're talking about young people. I mean, they are also very seriously considering climate problem, and then that's important problem, and then they working very hard. And then I believe this problem so complicated, climate problem, so complicated, very challenging, but I believe we will solve the problem together in the future. And then with you, your vision, and then your student implementation, and we'll do that for sure. I very optimistic. Thank you. I have some more question, but I want to check with the other people's question. Okay, this is not here. Okay. I guess your talk is perfect in that case, so everything's clear, super clear. Okay, and then now I see my time is just almost 11, so I probably need to conclude, and then we continue to chat.
Sure.
So thank all for joining and staying today. I believe everyone very busy with the new semester start, but still we have some people over here. So we'll meet again next week on September 8th, Thursday, the same time, the same Zoom link. We'll have Professor Julius Lux from Northwestern University, one of my favorite, colleagues, and also Dr. Boya Wang from University of Texas at Austin. As usual, the follow-up informal chat will occur without recording. So please stay here if you are interested in chatting with today's speakers. I will promote you to panelists who can show your handsome and pretty face, and then you can speak. So thanks. I'll stop recording. Just give me 1 second.